Hi, everyone. My name is Eryk Kropiwnicki. I'm a bioinformatician in the Mayan lab, and I'm going to be talking about Appyters and the Appyter framework that's being developed by the Mayan lab that allow you to create web applications for reproducible bioinformatics workflows. So what exactly are Appyters? At their core, Appyters turn Jupyter notebooks into their own functional standalone web applications. And so each Appyter first presents a user with an input form that can be modified. And there's like an example on the right there from the enrichment analysis visualization Appyter, where a user can paste their own input gene set and select an enricher library of choice. And once they submit the input form, they'll retrieve visualizations of the top results from gene set enrichment analysis. And so this is sort of just one example of an Appyter, and that possibilities are endless. Appyters allow those with programming experience to create many of these kinds of web-based reusable workflows. And these include already existing applications that are available and have been built by the lab, some of which were customized machine learning pipelines, Appyters that analyze omics data, and other Appyters that produce publishable figures. And sort of the key point here is that, and I'll stress this throughout the presentation, is that Appyters are really easy to create. They require much less overhead and less skill than traditionally developing web applications. And furthermore, these workflows that are created by us in the community are available online for those that don't have very much experience with programming and who need to accomplish some sort of computational task. So individual Appyters, when they're accepted through a GitHub pull request, are ingested into the Appyter catalog. And so again, if someone has like a specific bioinformatics workflow in mind, they can browse the catalog by specific tags or keywords to find what they're looking for. And currently there are about 97 Appyters in the catalog, and that number is continually growing as the lab contributes more and more of these Appyters. So the Appyters and the Appyter framework was developed by Daniel Clark in the Mayan lab. And here's sort of the general overview for creating an Appyter. So at the lowest level, each Appyter must begin as a Jupyter notebook that accomplishes some sort of computational task. And for those unfamiliar, the Jupyter notebook provides like a interactive computational environment where it incorporates code, markdown text, and ways to sort of visualize generated figures in the browser, and sort of transformed the way we can communicate these data analysis pipelines in a reproducible way. And so Appyter sort of rests on top of these Jupyter notebooks. And so the Appyter software development kit can be loaded into the Jupyter notebook and allows for the addition of this meta programming aspect that adds these input form options. And depending on these input form options, variables and other logic conditional on these options will be assigned and executed when the notebook is launched. And once an Appyter notebook is executed in the cloud, with a particular set of input form options, if these same options are submitted again, the user will be redirected to a persistent Appyter instance. So these are readily shareable workflows as well. So just to provide an example, here we have an Appyter notebook that is simply adding two numbers that are input by the user. And the first cell contains the code that's required to initialize the Appyter SDK. And then in the second cell, we have this %%. These are referred to as Jupyter magics. %% Appyter code exec line, which allows for the instantiation of Appyter functionality in this particular cell. And the variable assignments here they have this double bracket syntax, which is Jinja 2 syntax, which is a widely used templating programming language. And so anything inside the brackets is going to be evaluated by the template system to construct the actual source code at runtime. So this allows for the assignment of these input fields. In this case, an int field, an integer field, which includes a dictionary of information that you can assign. For example, like the default value that's presented to the user when they first open the input form. And so Appyters also come with a wide variety of other input form options, including file fields, drop-down menus, Boolean fields, and others, which allow for further customization of the Appyter notebook when it's executed. So num1 and num2 here are global variables that are initialized by the input form elements, and they can be used while performing other operations in subsequent cells, such as printing the summation of those two variables. So the Appyter package also includes a CLI, command line interface, for debugging and executing Appyters. So to launch an Appyter locally, you simply input the statement shown here in this black bar. This profile tag decides how the input form is stylized. The default we have is BioJupys, but there's other custom profiles that can be added by you to change the look and feel of the input form. And lastly, in this CLI statement, you also have to pass the file name of the Appyter notebook to be launched. So then the Appyter is launched locally and can be tested in the browser. And the input fields, of course, can be altered. And when submit is pressed, the notebook is executed. And so for our example Appyter here, we see that we assign num1 to 1, num2 to 2, and then we print the addition of those two variables. So this was a pretty bare bones sort of hello world example of what Appyters are capable of. And in the subsequent slides, I'm going to show some examples of more sophisticated Appyters that are currently available in the Appyter catalog. So Dr. Minji Jeon, who introduced herself before, is a postdoc in the Mayan lab, and she created an Appyter for the processing and analysis of bulk RNA sequencing data. So the input form contains options for uploading data and metadata. Here, you could choose the class columns and the control labels. Mousing over these tooltips gives a better description of each field. So you could select these normalization methods, ways to visualize the omics data, as well as lastly, options for how differential gene expression is calculated and selection of various analyses. So once the input form is submitted, the Jupyter notebook is executed with the selected input form options. And there are options at the top also for working with the notebook locally or downloading it, and an option to toggle the code if you're only interested in the analyses and the output figures. And so as you scroll through the Appyter, you can see the various sections dedicated to each analysis that you could actually traverse by clicking on each of these in the table of contents. And so each of these sections contain visualizations of the results. And there are also download links to publication-ready figures. So as you scroll through, you can start to see how one can create an entire workflow and allow users the ability to bring their own data and tailor their analyses with the available options. So next is the PRISM X Appyter, which was created by Dr. Alex Lockman in the lab. And this is a machine learning Appyter for predicting gene function using gene co-expression correlations from RNA-seq data sourced from ArchES4. And so 50 these correlation matrices are pre-computed and stored in the cloud. and at runtime, the correlation data is extracted from the cloud, and it's used with a pre-trained random forest model. The random forest model is applied on these features to rank all the gene sets from a user-specified gene set library. And so here it is in action. So the input form includes an option for inputting a valid gene of interest and gene set library to make predictions from. And then when you submit the input form, we're once again greeted with the Jupyter Notebook output. And then scrolling through, we can see the data frames of the correlation matrices, which you can inspect, see their structure, a ROC curve, which displays the performance of the model, as well as lastly, a downloadable table of predictions. In this case, the table is displaying Go biological processes that map K1 was not previously annotated with. And we have a third Appyter here created by Kerwin Chen, who is a recent undergraduate summer trainee in the lab. And his project was focused on analyzing the differential expression of a user-selected gene across several signatures from Geo. And so in this case, we're looking at the gene AKT1, and scrolling through the notebook, there's first a volcano plot, where each point represents a signature from Geo. Usually volcano plots show the differentially expressed genes, but in this case, it's signatures. And they show the p-value, fold change. And there are also two tables listing the top and bottom 10 signatures ranked by p-value, and a link to the Geo study from which the signature was collected. And also, if there is a orthogonal gene in mice, the same analysis is repeated, except for mouse studies from Geo. So ultimately, the work we do during the hackathon will culminate in an Appyter. And Appyters are, they're capable of being Dockerized and deployed, and they can run anywhere. And but to be in, if you work on an Appyter and you want to submit it to the Appyter catalog, to be ingested into the catalog, a pull request must be submitted on GitHub for the Appyter in question. The directory must be structured in a particular way in order for your submission to pass the GitHub tests and the Appyter to be included in the catalog. And so an example of an Appyter directory structure is included in GitHub, and key items include the notebook itself, obviously, any package dependencies to allow for Dockerization, and importantly, a appyter.json file, which is a JSON schema validated descriptor file that enforces essential information to ensure Appyter fairness and for also displaying this metadata in the catalog. So that about sums up the crash course on Appyters. So we will be eventually developing one of these Appyters ourselves. And so far, the ideas that have been discussed are a knowledge graph that connects, predicts and visualizes gene disease and drug associations. Another suggestion was machine learning, a machine learning pipeline to predict gene functions specifically for IDG targets. And I suppose after my presentation, we could take some more suggestions or have a discussion, unless there's a time slotted off for that later on, in which case we could talk about that then. I've also included a slide of resources to refer to for anything related to building Appyters. And Anna, I suppose, will disseminate these slides, or I could upload it to Google Drive. And lastly, we want to thank the NIH for supporting this work. And I'm looking forward to working with everyone over the next few days. That's it for me.
Thank you, Eryk.